Greetings everyone. Welcome to week 7 of our introductory animal physiology course. Myself Dr. Tulasi Raman, working as an assistant professor in the Department of Veterinary Physiology and Biochemistry, Faculty of Veterinary Animal Sciences, Institute of Agricultural Sciences, RGSC, Banaras Hindu University. This week we are beginning an exciting new module on the excretory system. In our first lecture, titled Nephron, Functional Morphology, Urine Formation, we'll explore the structure and function of the nephron, the core unit of the kidney. Today, we will examine how the nephron filters blood, forms urine, and play a vital role in maintaining internal stability or homeostasis. By dividing into these details, we'll gain a better understanding of how animals adapt and regulate their internal environments. I am looking forward to engaging with you all on this important topic, and I am here to support your learning journey. Let's begin. So, the Claudius Galen, he is the first scientist who demonstrated that urine was formed in the kidney, not the bladder. Before, it was thought that the urine was formed in the bladder but not by the kidney. Later, the Arthur Cushney, he is another important scientist in the renal physiology who proposed the modern theory of the secretion of the urine, which we will see in the upcoming slides. So, what are the functions of this renal system or the kidney as such, if you say? The first and foremost function is that excretion and elimination of the various waste products which is secreted from the body fluids such as urea, creatinine and uric acid. Apart from that, it also excretes and eliminates the foreign chemicals that are present in the body. And the next important function is that the homeostatic regulation. That means it is trying to maintain or regulate the arterial blood pressure as well as the volume of the body fluids, especially the blood. So, such as with the help of the water salt balance and the acid base balance. And the third important function of the kidney is that endocrine functions, such as by secreting the renin angiotensin system hormones as well as the various other humoral factors such as erythropoietin as well as 1,25 dihydroxycholecalciferol, that is the active form of the vitamin D3. And the last one important function of the kidney is that it involves in the gluconeogenesis process. So, gluconeogenesis process is nothing but it is the derivation of the glucose from the non-carbohydrate sources such as lactate, etc. So, these are some of the important functions of the kidney. Now, as we said, the kidney, who is the key player of the kidney is that nephron. Nephron is the basic structural as well as the functional unit of a kidney is nephron. You should not forget this. And this nephron number varies from animal to animal. But this is not the case in the human, whereas in case of the animal physiology, the nephron numbers varies from species to species. Such as in cattle, it is around 40 lakhs nephrons per kidney, whereas in pig it is 12 lakh 50,000, whereas in dog it is 4 lakh 15,000, and in cat it is 1 lakh 90,000, and in humans it is around 10 lakhs nephrons per kidney. Let us come to the types of nephron which is present in the animals. So, the nephrons are classified based on the location of the glomeruli, and the next important criteria is that how far or how long the nephron penetrates. So, based on that, the nephrons are classified into cortical nephron as well as the juxtamedullary nephron. If you see the cortical nephron, this is the structure of the cortical nephron, where you can see this is the glomerulus, and you can see the extent of the loop of Henle. It is just extended up to the dorsal medullary part of the kidney, right? This is the dorsal medullary part. Whereas if you see the juxtamedullary nephron, where this is the glomerulus, and see the how extent this loop of Henle is extended. It penetrated deep into the medullary part. So based on this, the nephrons are classified into cortical nephron as well as the juxtamedullary nephron. Let us come to the type of kidneys. So these kidneys are broadly classified into two types in the animals. One is unipyramidal and the next one is that multipyramidal. So unipyramidal kidneys, this is present in the species such as small ruminants, carnivores, and then horse. In this species, you will find the unipyramidal kidney. So this is unipyramidal kidney which is having a single pyramid. It does not have a multiple pyramids. That means multiple papillae is absent. Whereas if multiple papillae or multiple pyramids are present, then that kidneys are called as a multipyramidal kidney, which is normally present in the large ruminants and then in pig. So in this species you will find the multipyramidal kidney. Let us go into the structure of the nephron. So the nephrons, it is containing, the first important part is the glomerulus, which contains the glomerulus capsule. The capsule contains the tough capillaries which is present inside the capsule. You can imagine the glomerulus capsule is nothing but a T sieve. It looks like that only. And then this glomerulus capsule will be continued with the proximal convoluted tubule followed by the proximal tubule, straight tubule, and then that will get continued into the loop of Henle. So this loop of Henle have two parts. One is ascending as well as descending. So descending means it will move from the top to the bottom, whereas ascending means it will move from the bottom to the top, right? So the proximal straight portion, from there it will continue as a thin descending limb, which is followed by the thin ascending limb followed by the thick ascending limb of loop of Henle, and it will continue as a distal tubule, the distal convoluted tubule, and the later on it will go as a collecting tubule or the collecting duct, you can say. So in the collecting tubule, it will be classified into cortical collecting duct and then medullary collecting duct. And one important another structure is that the papillary duct are also called as duct of Bellini. So this is about the overview of the nephron structure. If you see the glomerulus, as I told you already, so this glomerulus having a capsule, this is glomerulus capsule, and which contains the tough capillaries inside, and these capillaries have some tubules, right? On the tubules, on the side of the tubules, you will find the podocytes. Podocytes, why the name given? Because it is having some foot-like processes or the projections. Due to that reason, it is given as a podocytes. Apart from that, it is having the mesangial cells. Again, the mesangial cells, there are two types of mesangial cells. One is extraglomerular mesangial cells and intraglomerular mesangial cells. So with that, we just saw what is a nephron, what are the different types of kidney, what are the different types of nephrons, and how it differs from one species to another species, how much nephrons will be present in different animals. Now we'll see the next important aspect of in the kidney itself is that juxtaglomerular complex. So juxtaglomerular complex, as I said, it is a complex. That means it involves more than one or two structures. That is why it is called as a complex. So this complex contains three important structures. First one is that macula densa, and the second one is that JG granular cells, and the third one is that extraglomerular mesangial cells. It is also called as Lasix cells. So these are the three important structures which involves in the juxtaglomerular complex. So let us see this, how this juxtaglomerular complex is formed. So when you see the distal convoluted tubule, when this distal convoluted tubule comes towards the afferent arteriole, come in contact with the afferent arteriole and efferent arteriole, where there are some of the epithelial cells will get modified so into a thick granular cells, and these cells are called as macula densa cells. This is a modified epithelial cells which lines the distal convoluted tubule, where it will come in contact with the afferent arteriole and afferent arteriole. Other areas you may not find this macula densa cells except the distal convoluted tubule where it come in contact with the afferent arteriole and the efferent arteriole. Except that area you will not find this cell. The next important is that there is a space between the macula densa cells and the space and the afferent arteriole and the efferent arteriole. You can see here there is a space. So in this space one another type of specialized cells are filled. So these specialized cells are called as mesangial cells or lattice cells. Already I told you about the mesangial cells. Now these mesangial cells are located outside of the glomerulus. Due to that reason it is called as a extraglomerular mesangial cells. So these extraglomerular mesangial cells are present in the space between the macula densa cells and then the afferent arteriole and the efferent arteriole junction. Then the afferent arterioles where the come in contact with this macula densa cells and the lattice cells where the cells are get the endothelial cells are get modified into a granular cells. It contains some of the granules in it. So these cells are called as granular cells here. This area you will find the another specialized cells called as the granular cells. So these granular cells contain the granules. Which granules? The proteolytic enzyme the renin has synthesized and stored in the form of the granules, and when the stimulus comes then automatically the granules get released into the system, I mean in the blood. Okay. So this is about the juxtaglomerular complex. So three important structures involved, and what are those, how it is involved, that and all you have seen. Then next we will see the urine formation. So when you say urine formation, so urine formation involves three important processes. The first one is called as glomerular filtration. Then second one is the tubular reabsorption, and the third one is tubular secretion. These are the three aspects. So the first is that when any liquid is formed, the first is that I told you, you imagine the glomerulus as a sieve. So you will just pour something in that sieve, then the sieve will get filtered everything and it will get collected in any container. Okay, the same process also performed by the glomerulus in the kidney. So what is this glomerular filtrate? This glomerular filtrate is nothing but an ultrafiltrate of plasma. So ultrafiltrate of plasma means it is devoid of the cells as well as the other formed elements, and then the some of the particles which are anionic in nature. So because this glomerular membrane is aligned with the polyanionic property, so due to that reason the anionic nature of any complex cannot pass through this membrane. Okay, so the glomerulus, it is made up of three important structure. The first is that the capillary endothelium. Endothelium means usually it is present in the blood vessel. Okay, then there will be a basement membrane. Okay, then third important structure is that the glomerulus endothelium. Okay, so these are the three important structures involved in the glomerulus. So any compound or any particle has to pass these three structures, and then it will enter into the glomerulus. So when it enters, then it will be called as a filtrate. So what type of filtrate it is? It is an ultrafiltrate. It is also equivalent to the plasma of the blood. That means the same thing is present here except the blood cells as well as the other some proteins and all. Fine. So this is about the glomerular filtrate. Now in this glomerular filtrate itself, you must know about certain terminologies such as RBF and then RPF. So RBF is nothing but the renal blood flow, and then RPF is renal plasma flow. Renal blood flow means it is the rate at which the blood flows to the kidney, whereas renal plasma flow is the rate at which the plasma flows to the kidney, right? In addition to that, it is the other terminology such as FF, that is filtration fraction. So in addition, and then GFR is the glomerular filtration rate. That means it is a rate at which the amount of blood will enter and then the filtrate formed. So that is the glomerular filtration rate, GFR. So this glomerular filtrate, how it will be formed? All these filtrates are formed with the help of some pressure. Okay, so there are two important pressures is involved here. The first one is called as a hydrostatic pressure, and next important pressure is that colloidal osmotic pressure, you can say oncotic pressure. So these are the two important pressures. So the hydrostatic pressure is the pressure which is exerted to move the water from inside to outside, whereas the colloidal osmotic pressure or the oncotic pressure, it will try to pull the water inside. Okay, so one is trying to push the water and another one is pulling the water or retaining the water with them. So that is the main difference between this hydrostatic pressure and the colloidal osmotic pressure or the oncotic pressure. So the first one is that the glomerular capillaries, these glomerular capillaries is also called as high pressure system. High pressure system means it will just try to favor the filtration process. That means it will try to push the molecules as well as the water from this part to the another part. Okay, so that is a high pressure system. Then the next low pressure system also exists in the same kidney, in the glomerulus, that is made by the peritubular capillaries, where this is a low pressure system that favors the reabsorption of the solutes as well as the water molecules. So these are the two important pressure systems which is present in the glomerulus. Now if you see the hydrostatic pressure in the glomerular capillaries, which will favor the filtration, which comprises of around plus 60 mmHg, the pressure, whereas the colloidal osmotic pressure, which of the plasma proteins, especially the albumin, which contributes for the oncotic pressure of the plasma, which is against the filtration process, so that gives around minus 32 mmHg, whereas the third one, the hydrostatic pressure of the filtrate. So this is the glomerulus, for example, this is the glomerulus, here the filtrates are there, whereas this is the blood part, right? So one is that it is favoring, another one it is against, and the third one is against, it is against. So if you see the hydrostatic pressure in the filtrate formed, so that is also against the filtration process, so which will contribute on minus 18 mmHg. So as a result, the net filtration pressure created in the kidney is around plus 10 mmHg. So ultimately anyway filtrate will be formed. That means some 60 ml of blood is coming, 10 ml of blood will be filtered, because other than that minus 32 ml pressure is against the filtration and minus 18 ml pressure against I'm just giving in terms of ml so that you can able to understand, that's why, but it is not ml in terms of this one. Now let us see what are the factors which favors this glomerular filtration rate. The first is that the vasoconstriction and dilatation of the afferent arteriole and afferent arteriole, this play a major role in the either the increased or reduction of the glomerular filtration rate. So the first we will see what are the criteria or the factors which is responsible for the decrease in the GFRs, the glomerular filtration rate. So the first is that the constriction of the afferent arteriole. So to understand this, for example, the afferent arteriole, you can remember this value 100, whereas in the middle it will be again 100, then in the afferent arteriole it will be 100. Remember, these are the three numbers. You just remember in terms of this number so that it will be easy for you to understand. So if there is a constriction at the afferent arteriole, this 100 will become 50, for example. Okay, then what will happen in the middle? In the middle also it will become 50, right? Then once the entry also less, and then even in the glomerulus also less amount of blood is there, then obviously you can expect a less GFR only, right? Whereas in the other case, if you take where the dilatation of the efferent arteriole is taking place. That means the exit point is getting dilated, but not the entry point, as well as inside the glomerulus there is no change. That means entry is 100 and even in the glomerulus it will be 100, but the exit is 200. So in this case what happens? As soon as the blood enters, it will not give enough time to stay in the glomerulus. As a result, it will exit. So due to that reason, the glomerular filtration rate also decreases, right? Then let us go and see some scenarios where the glomerular filtration rate increases. So here you can see the constriction of the efferent arteriole. So that means again you can see here 100, right? Here again it will be 100, but here it is 50. So in that case what happens? Only 50 is going out instead of 100. So that means some other 50 stays here itself, right? So due to that reason, more blood is getting accumulated in the glomerulus itself. As a result, it will give more time for the filtration. So the GFR get increases. And then next scenario is that dilatation of the afferent arteriole. When the entry here itself is instead of 100, it enters 150, but the glomerulus it is only 100 and then the exit is another 100. That means entry is more. So as a result, but the exit is same. So again more blood is stayed in the glomerulus itself. As a result, it will be filtered. The glomerular filtration rate increases. Now what are the other determinants which play a major role in increase and decreases of the GFR? That also we will see. The glomerular filtration coefficient, the KF, which will have a directly proportional relationship with the GFR, so it increases, GFR also increases, right? Whereas Bowman's capsule hydrostatic pressure, it is having a inversely proportional relationship. That means if it increases, then it decreases the GFR. Then glomerular capillary colloidal osmotic pressure increases, then again it decreases the GFR. Then the glomerular capillary hydrostatic pressure, then systemic arterial blood pressure, and then efferent arteriolar resistance. These all three are having a directly proportional relationship. That means if these increases, then these three will decreases the these three will increases the GFR. Then the afferent arteriolar resistance, if it increases, then it decreases the GFR rate. Fine. Now what are the different hormones involved and how it influences the GFR, that also we'll see. The epinephrine and norepinephrine, right, which is a stress hormones that increases, that main function is to increase the arterial pressure, that you know, and then it is having some direct action on the kidney, right? So there are two actions it will produce in the GFR. One is indirect action, another one is direct action. So indirectly, if you see, it increases the GFR provided the BP held constant. Otherwise what will happen? It decreases the GFR. Whereas the endothelin, if you take endothelin as well as the angiotensin 2, both of them it causes the vasoconstriction, but the angiotensin 2 is a potent vasoconstrictor. When if someone asks what is a potent vasoconstrictor means, then you have to go with the angiotensin 2 instead of endothelin, right? Both are causing the renal vasoconstriction. As a result, it both decreases the GFR. Whereas nitric oxide as well as the prostaglandin and prostacyclins, the autocoids, again it causes the vasodilation. As a result, it increases the GFR. Then bradykinin also, it causes vasodilatation, causes the increase in the GFR. Now whenever the blood pressure changes, maybe 60 or 80 to 120, if it is maintained in the normal range, there is no problem. But if there is a mild change within this limit or just going beyond this limit up to certain extent, then the kidney itself will perform certain type of autoregulatory mechanism to bring it back to the normal range. So that type of mechanism is called as autoregulatory mechanisms. There are two important autoregulatory mechanisms in the kidney. The first one is that myogenic mechanism. Myogenic means it is related with the muscles. So whenever there is a pressure increases, BP increases, then that causes stretch of the arterial walls. Arterial walls get stretched. As a result, the pressure receptors which is present in the arterial walls will sense that one and it will send a signal, right? So that is the myogenic stretch receptors. That's why it is a myogenic stretch receptors, causes the opening of the voltage gated calcium channels, causes the influx of these calcium channels, calcium from the extracellular fluid to the vascular smooth muscle cells, causes the vasoconstriction. So that results in the decrease in the GFR. So whenever increased blood pressure causes the increased GFR, so then the autoregulatory mechanisms will come into play, and that triggers the myogenic stretch reflex. Then the next important autoregulatory mechanism is the tubuloglomerular mechanism or the macula densa feedback mechanism. So whenever the arterial pressure decreases, that will be having the decreasing the GFR also. As the GFR decreases, so less sodium chloride will be filtered, so that will be sensed by the macula densa cells, sends signals to the granular cells, JG granular cells, that releases the renin. So the renin will in turn triggers the angiotensin II production. As a result, it increases the efferent arteriolar resistance. Then it will have a negative effect, and then like that it will try to maintain the homeostasis. Then let us come to the tubular reabsorption. So this is about the glomerular filtration. Once the filtrate is formed, and then it goes from the glomerulus to the tubules. When it enters into the tubules, the proximal tubule, then this will be called as a, not a filtrate, it is a tubular fluid. From now onwards, this fluid is a tubular fluid. You should not use the word filtrate, right? The most important part in the tubular fluid is the renal clearance. So renal clearance is nothing but the capacity of the kidney to clear any substance from the plasma. So that is the renal clearance. Mostly it will be calculated with this formula, the Cx. X means any compound, you can say. C, the clearance of a substance, any X substance, it will be expressed in terms of Ux, that is the concentration of that substance in the urine multiplied with the rate of the urine formation, which is expressed in ml per minute, divided by the plasma concentration of that particular substance. So this is how you can calculate the renal clearance of any substance. So for example, if in case of the creatinine as well as the inulin, the GFR is almost equivalent to the clearance. That means 100% it will be cleared in case of creatinine as well as inulin. Whereas in case of the electrolytes and urea, if you take, the clearance is lesser than the GFR. That means 100 molecules in GFR it produces, whereas 80 or lesser than 100 only it will get cleared. Whereas in the nutritional substances such as glucose and amino acids, where the clearance limit is 0, that means 100% it will get reabsorbed. Whereas in the organic acids, bases, potassium and hydrogen ions, if you take, the clearance is more than the GFR. That means here the secretion is happening, tubular secretion, right? So that is why it is 100 in GFR, it is filtered, but whereas it is excreted 150, that means more is filtered. Now, what are the mechanisms which is involved in the tubular reabsorption is that the first one is that in the proximal tubule, 3 important mechanisms are there. The first one is that the sodium co-transported with the amino acids or the glucose. Okay, so this is one important mechanism. The next important mechanism is the sodium counter transported with the potassium ion, hydrogen ions, because whenever the hydrogen ions inside the cell increases, it decreases the pH. That means the acidity of the cell rises, right? So no one will try to live in an acidic environment. The same way the cell also does, it excretes the hydrogen ion in exchange of the sodium ions. Then the third one is chloride driven sodium transport. So sodium and then chloride. So chloride play a major role because it concentration is present in outside. So due to that reason, the chloride drives the sodium to enter inside the cell. So these are the 3 important mechanisms you can see in the proximal tubule for the reabsorption process. Whereas coming to the loop of Henle, ascending thick limb of loop of Henle, if you take, there you will find the sodium potassium 2 chloride co-transport mechanism. So sodium and then potassium and then chloride mechanism. So this is a co-transport again. Then the last place where the tubular reabsorption used to take place is the collecting duct, where the conductive sodium channels are there. So the sodium channels, always remember, these all are taking place from the tubular fluid to the interstitial fluid, because the concentration is higher, these ions is in the tubular fluid than the interstitium. So it moves from higher concentration to the lower concentration. That is why these are all the co-transport or counter transport without any ATP is involved. If ATP is involved, means it is going against the concentration gradient. Please remember that one. Then the last thing you need to remember here is the Tmax, that means the transport maximum. There is a maximum limit for everything in the body, right? Even for us also, the same way, the kidney also having a maximum limit for any compound it will be excreted. So for glucose it will be 375 mg per minute. For amino acids it is 1.5 millimolar per minute, where plasma proteins it is 30 mg per minute. Now the third aspect of the urine formation is the tubular secretion. So whatever is not required for the body, that will get excreted, or whatever is in excess, that also will get excreted, right, or secreted you can say. So the first important is that H plus ions. So these H plus ions, how it will be formed inside the cell is that, first of all, the carbon dioxide is generated as a result of the respiration, cellular respiration. It will react with the water, and then it forms the H2CO3 carbonic acid, that will give rise to H++ bicarbonate ions. So these H+ ion concentration keep on increasing if the carbon dioxide is accumulating, right? So as a result, the H+ need to be secreted out, right? So the H+ secretion is mostly takes place in the distal nephron, right? And then the bicarbonate chloride exchange mechanism is another important mechanism, where the bicarbonate ions will enter and instead of the chloride ions. So it is an exchange will takes place, bicarbonate as well as the chloride, depending on the requirement. And the next is that the potassium ions, usually the tubular secretion, the secretion of the potassium ion takes place at the proximal tubule, loop of Henle, as well as the distal part of the nephron. And then the urea, if you take, it is mostly seen at the distal convoluted tubules, whereas in the proximal convoluted tubule you can see the secretion of the creatinine and the antibiotics, diuretics, and the uric acid. So these all components are secreted even the proximal convoluted tubule itself. And coming to the again the renal clearance, I told you for para-aminohippuric acid, the renal clearance is that limit is at 80 mg per minute, whereas creatinine it is around 160 mg per minute. Now the hormones which involves in the which having an influence in the urine formation, that we'll see. The first is that aldosterone, it's a hormone, right, which the site of action is that collecting tubule and duct. Most of the function includes, it increases the NaCl concentration, right, and then water reabsorption. Then it increases the potassium secretion. Remember, the aldosterone's main function is that increase reabsorption of the sodium chloride. Remember, the sodium is having few lovers. So the first lover of the sodium is the chloride, and the next lover is the water. So whenever the sodium will get reabsorbed or secreted, along with that chloride also, along with that water also will get secreted or reabsorbed. So don't forget that one. So sodium's two lovers. So whenever the sodium is reabsorbed, chloride and water also will get reabsorbed. Instead of that, it is secreting the potassium ion. So higher potassium concentration is one important stimulant for the secretion of the aldosterone from the zona glomerulosa. Then angiotensin 2. Angiotensin 2 mostly it acts on the proximal tubule, thick ascending loop of Henle, and the distal tubule and collecting tubule, where the important function is that again it increases the sodium chloride water reabsorption, I have told you, right? Then it increases the hydrogen ion secretion. Remember, the aldosterone causes the potassium ion secretion, whereas the angiotensin 2 causes the H+ secretion. That is the most important and the important difference between these two. Then ADH, the antidiuretic hormone or the vasopressin you can say, it act on the distal tubule or the collecting tubule, where it increases the water reabsorption. It is an antidiuretic, that means against the diuretic process. We will see what is the diuresis in the next lecture, right? Then atrial natriuretic peptide, it will act on the distal tubule or the collecting tubule, where it decreases the sodium chloride reabsorption. So as a result, more sodium chloride will be excreted along with the water. Remember, wherever the sodium chloride is leaving, then it will take the water also along with that. Then parathyroid hormone, it will act on the proximal tubule, thick ascending loop of Henle, or the distal tubule, where it causes the reabsorption of the, decrease the potassium reabsorption, right, and then increases the calcium reabsorption. So the most important is the parath hormone is that increases the calcium reabsorption. Why it is increasing the calcium reabsorption? Because you know the parath hormone involves in the calcium homeostasis. You might have learned that, right? So parath hormone, whenever it comes, it is related with the calcium. So that's why it increases the calcium reabsorption, and then it excreting the potassium ions, phosphates. So with this today, what we have seen is that what is a nephron, right? And what are the different types of kidney and the different types of nephron, right? Based on the glomerulus position as well as the extent of the loop of Henle into the medulla. And then different types of kidney and the nephron and its structure, what are the different structures it is involved, and then how it is made, and then different autoregulatory mechanism. Two important autoregulatory mechanisms we discussed today. And then the urine formation, the first process is that glomerular filtration, and the second is that in this glomerular filtration, JJ apparatus also we have seen, right? And then what is renal clearance, then what is tubular reabsorption, what are the different mechanisms at the proximal tubule, loop of Henle, distal tubule, and then the tubular secretion, where the different secretion will take place at the different levels of the nephron, and ultimately the hormonal control of the urine formation in as a summarized one. So with this, I'll end today's topic, today's lecture, where we discuss the total nephron functional morphology and urine formation. And in the next class, we'll see the countercurrent mechanism, the RAS mechanism, as well as the diuretics and its classification. Thank you.